immunotherapy.
Yeah.
I mean, it really holds this incredible promise, right? A durable, maybe even personalized cure.
Absolutely.
And for those patients whose cancers do respond to things like immune checkpoint inhibitors, the PD-1, CTLA-4s, the results can be just revolutionary.
Life-changing.
But, and this is the big but, the painful question that really drives so much of oncology today is, why does this miracle work? Wonders for one person, maybe even cures them, but then fail completely for someone else. That massive variability. That's what we're diving deep into today.
And that variability, as you said, it's everything. It really comes down to the battlefield itself. The tumor microenvironment, the TME.
Okay.
And we generally split tumors into two main camps based on that environment. First, you've got the hot tumor. That's the goal.
Right.
It's naturally inflamed, packed with immune cells, lots of those killer T cells already there, ready to go. ICIs work well because the immune system's already primed, just kind of held back.
So the treatments just need to release the brakes, essentially.
Exactly, take the foot off the brake. But then there's the second type, the cold tumor.
The problem child.
The problem child, yeah. A master of immune evasion. It has hardly any T cells around, or maybe none at all, and the whole environment is just designed to suppress any immune response.
Like a fortress.
It is a fortress, totally resistant to detection. So our mission, really the field's mission, is figuring out how to turn these cold, hidden tumors into hot, inflamed battlegrounds that the immune system can fight.
Okay, let's unpack that. We need to dissect how these cold tumors build their defenses, this sort of three-pronged system they use to hide, suppress, and resist, and then look at how researchers are trying to, well, flip that switch, make them treatable.
Right. And to really grasp the challenge, researchers use something called the immunoscore system. It helps classify the geometry of these cold tumors. Think of it like mapping the security layout.
Okay, different types of fortresses.
Exactly. First, you have immune desert tumors. Basically, no T cells, not in the core, not at the edges, just empty. Immune surveillance is completely absent.
The guards aren't even near the building.
Not even close. Then, type 2, altered, excluded tumors. Here, the lymphocytes, the T cells, they are around, but they're stuck outside. They're confined to the stroma, the surrounding tissue, blocked by physical barriers. They can't get into the tumor core itself, like they're patrolling the outer wall but can't breach it.
Okay.
And the third type, altered immunosuppressed tumors. In this case, maybe some T cells got inside, but they're just not working right.
Handcuffed.
Kind of. Their numbers are low and they're functionally impaired. Maybe by chemical signals like TGF-beta or by other suppressive cells hanging around, like TREGs.
And are there common features to all these cold types, besides just not enough working T cells?
Yeah, they share some key traits, usually a low number of mutations overall, which means fewer weird proteins or antigens to spot.
Less for the immune system to recognize.
Right. They often have problems with their antigen presentation machinery, the system for showing those antigens. Plus, dense structural barriers, like we mentioned, and really chaotic abnormal blood vessels. All this contributes to blocking that fundamental process, the tumor immunity cycle.
Ah, yes. The cycle. That's the sequence the body needs to follow for a successful attack, right?
Yeah.
Let's walk through that, because the tumor seems to fight it every step of the way.
It does.
Step 1. The tumor has to release antigens, those flags that say, I'm abnormal.
Correct.
Step 2. Specialized immune cells, dendritic cells, or DCs, have to capture those antigens.
The scouts.
The scouts, right?
Right.
Third, the DCs travel to the lymph nodes and basically train up naive CD8 plus T cells, the killer cells, the CTLs.
Priming the soldiers.
Fourth, those prime CTLs have to navigate back to the tumor and actually get inside the TME.
Infiltration.
And finally, step 5, the CTLs have to recognize the tumor cells via MHC and eliminate them.
The kill step.
And the research we're looking at today uses this really useful framework, the 3Cs model, to categorize how tumors break that cycle at every point.
Exactly. Camouflage, coercion, and cytoprotection. Those are the pillars of cold tumor defense.
Okay, let's tackle the first C, camouflage. This is the hiding part, right? Making the tumor invisible or inaccessible. How does it put on the invisibility cloak?
Well, a major way is defective antigen presentation. Tumors need to show those antigens using MHC class I molecules.
The ID display system.
Right, but they actively sabotage it. One way is genetic evasion. They get mutations in key parts of the MHC-I machinery, like a protein called beta-2 microglobulin, or B2M. If B2M is mutated, which happens a lot in lung cancer, for instance, the whole MHC-I complex just can't assemble properly. The tumor cell basically deleted the molecular slot where it's supposed to show its ID.
So the T cell comes along and there's just nothing there to check.
Precisely. But they don't just break the machinery, they can also hide it using epigenetic silencing.
Molecular graffiti, you called it.
Yeah, that's a good term. They use enzymes, like EZH2, to add repressive chemical tags to the DNA that codes for MHCI components. This basically shuts down the production of those ID displays, locks the invisibility cloak on.
Wow, okay, so the tumor's hidden. But even if T cells could see it, they need those dendritic cells, the scouts, to raise the alarm first. How does the tumor stop that initial engagement?
The impaired DCT cell crosstalk. The tumor releases chemicals, TGF-beta again, and lactate, which we'll come back to, that block the signals needed to recruit the right kind of DCs, the crucial CDC ones.
It jams the recruitment signal.
It does. And even if some CDC ones get there, the tumor cells hoist up "don't eat me" signals. The famous one is CD47. It binds to a receptor called SIRRP alpha on the DCs and basically tells them, nope, nothing to see here, move along.
Like flashing a badge that says friendly, even when it's not.
Exactly. It prevents the DCs from engulfing the tumor cell or its antigens.
Okay, so the immune system's being blinded, confused.
Yeah.
But let's say a few T cells do get primed somehow. They still face the fortress walls, the physical barriers.
And these are formidable. First, you have the abnormal tumor vasculature, or TAV. Tumor blood vessels are often a mess, leaky, twisted, poorly formed. This creates high pressure inside the tumor and low oxygen hypoxia. Both are really bad for T cells. They can't survive well, and they can't even stick to the vessel walls properly to get out into the tissue.
What stops them sticking?
Often signaling pathways involving VEGF disrupt the expression of adhesion molecules like ICM1 that T cells need to grab onto the vessel wall.
So the doorway's blocked, that's the first wall. What's the next layer?
That's the extracellular matrix ECM remodeling. You have these cells called cancer-associated fibroblasts, or CFs. They go into overdrive, producing tons of matrix proteins like collagen and creating this dense fibrotic barrier. They even use enzymes like lysyl oxidase, LOX, to cross-link the collagen fibers, making the matrix really stiff and tight. The pores can shrink down to maybe 50 to 200 nanometers. That's physically too small for a T cell, which needs more space to deform its nucleus and squeeze through. They are literally walled out.
Just physically blocked. Okay. And the last part of camouflage.
Yeah.
Messing with the map. Chemokine gradient dysregulation.
Right. T cells navigate using chemical trails, chemokines. Tumors mess this up in two ways. First, they suppress the chemokines that attract the killer T cells. For example, certain oncogenes like beta-catenin in melanoma can actively turn off the production of things like CCL4, a key T cell recruiter.
So they erase the come here signs.
Exactly. And second, they actively hijack the chemokine network to call in the wrong cells, the suppressive ones. Mutant P53, for instance, can make tumors pump out CXCL1, which recruits loads of neutrophils, and those often end up helping the tumor.
Wow. Okay, so camouflage is all about hiding and blocking access.
Yeah.
That brings us to the second C, coercion. It sounds more active, like suppression and sabotage.
It is. This is the tumor fighting back against any immune cells that manage to get close or get inside. It starts with building these immunosuppressive cellular networks.
We know the usual suspects, right? TAMs, TREGs, MDSCs.
The big three, absolutely. M2 like TAMs, the tumor-associated macrophages, TREGs, the regulatory T cells that enforce tolerance, and MDSCs, myeloid-derived suppressor cells. They all work to shut down T cells. But there are other players emerging too.
Oh, like who?
Well, one group getting attention is immunosuppressive erythroid progenitor cells, EPCs.
Red blood cell precursors. How do they suppress immunity?
Sometimes, often linked to tumor-induced anemia, the body starts making blood cells outside the bone marrow. It's called extramedullary hematopoiesis, EMH. These EPCs can end up in the tumor and release things like ARG1, arginase 1, and TGF-beta, which directly inhibit T cell function.
Okay, who else?
Tumor-associated neutrophils, TNs. We used to think of neutrophils mostly for bacterial infections, but in cancer, they can be bad news. They change their metabolism, produce damaging reactive oxygen species, ROS, and can form these physical shields called neutrophil extracellular traps, or NETs.
Nets.
Like
spider webs.
Sort of, yeah. Sticky webs of DNA and proteins that can physically shield tumor cells and also recruit more suppressive cells, like TREGs. It's a coordinated defense force.
That's incredible coordination.
Yeah.
And this ties into the next part of coercion, metabolic reprogramming, the Hunger Games in the tumor.
It really is like the Hunger Games. Tumors rewire their metabolism not just to grow fast, but specifically to starve and poison immune cells.
How do they starve them?
Glucose. Killer T cells need a lot of glucose to function. Cancer cells ramp up their glucose transporters, especially GLUT1, and just suck up all the available glucose in the TME. The T cells are left running on empty.
Okay, so they steal the fuel. What about poisoning?
Lactate. The byproduct of that rapid glucose consumption by tumor cells is lactate. We used to think of it just as waste, but it's a powerful immunosuppressor itself.
Oh.
It acidifies the environment, which T cells hate. And fascinatingly, recent work shows lactate can directly modify proteins on tumor cells, a process called lactylation. This modification can actually stabilize PD-L1, the don't kill me signal, on the tumor cell surface.
Wait, hang on. The tumor's waste product actively reinforces its own defenses.
Isn't that something? It's weaponizing its own metabolism.
Yeah.
Genius level evasion, as you said earlier.
That's wild. What else on the metabolic front?
Amino acid theft. Tumors guzzle amino acids T cells need, like glutamine, and they use enzymes like IDO1 or TDO to break down tryptophan.
Right, tryptophan.
They break it down into something called kynurenine. And kynurenine doesn't just starve T cells of tryptophan, it actively pushes other immune cells to become more suppressive tregs. It's turning the TME into a hostile soup.
Chemical warfare, basically.
Absolutely. And it doesn't stop there. They mess with nucleotides too, converting inflammatory signals like ATP into suppressive adenosine.
Okay, metabolic warfare is brutal. But then there's this whole other layer you mentioned, the neural immune axis modulation. The nervous system getting involved.
Yeah, this is a really hot area. We're realizing the nervous system is deeply intertwined with the TME. Take the sympathetic nervous system, the fight or flight response. When it's activated, maybe by stress, it releases norepinephrine. This binds to beta-adrenergic receptors, or beta-ARs, which are actually on CD8 plus T cells.
T cells have stress receptors.
They do. And when norepinephrine binds, it tells the T cell to basically chill out. It induces exhaustion, suppresses their killing function.
Which is why people are looking at beta blockers.
Exactly. Simple drugs like propranolol, a beta blocker, are being tested to see if they can block that signal and wake the T cells back up in the tumor.
Incredible. What about other nerves, like sensory nerves?
Yep, the sensory signaling pathway too. Nociceptors, the nerves that sense pain, release peptides like CGRP, calcitonin gene-related peptide. CGRP binds to its receptor, RMP1, also found on CD8 plus T cells. And guess what? It also causes T cell exhaustion and makes them less responsive to immunotherapy.
So both stress signals and pain signals are telling the T cells to stand down.
It seems that way. The tumor is exploiting these fundamental physiological pathways. It's like finding hidden control wires.
Okay, this is mind-blowing. The tumor is hiding, blocking access, poisoning the environment, starving T cells, recruiting suppressive allies, and telling the T cells to give up via the nervous system. But even if a T cell overcomes all of that and lands a hit,
there's still the third C, cytoprotection, resisting the kill shot.
How do they do that?
A few ways. One is immune synapse defects. For a T cell to kill, it needs to form a stable connection, the synapse, with the tumor cell to deliver the lethal payload.
Right.
Tumors can downregulate adhesion molecules, like ICM1 again, making it hard for the T cell to get a good grip. The connection is unstable, the payload delivery fails.
Make themselves slippery.
Kind of, yeah. They also mess with cell death signaling. Normally, when cells die certain ways, like necroptosis or pyroptosis, they release danger signals, dampiers, that alert the immune system.
The dying cell screams for help.
Exactly. Tumors don't want that. So they use epigenetic silencing again to switch off the genes needed for these noisy, inflammatory types of cell death, like RIPK3 or GSDMD. They try to die quietly, if they die at all. They also boost pathways that protect them from other forms of death, like ferroptosis, by upregulating enzymes like GPX4.
But the craziest one sounds like this ESCRT thing, instant repair.
ESCRT-mediated membrane repair. Yeah, it's pretty amazing. ESCOT stands for endosomal sorting complex required for transport. It's normally involved in fixing membranes inside the cell.
Okay.
But when a T cell punches a hole in the tumor cell membrane with proteins like perforin, or when pyroptosis tries to form a pore, there's a rush of calcium into the cell. This calcium influx acts like an alarm signal, triggering the ESCRT3 complex to rapidly assemble right at the site of damage.
Like a tiny emergency patch grip.
Exactly. It pinches off and removes the damaged bit of membrane, effectively sealing the hole within seconds or minutes. The tumor cell survives the attack and prevents the release of those danger signals. It dodges the bullet and muffles the gunshot at the same time.
That is just an unbelievable level of defense. Okay, so we've got the fortress map. Camouflage, coercion, cytoprotection. It seems almost unbeatable. Let's pivot. What's in the conversion toolkit? How are we fighting back?
Well, the key is realizing you can't just do one thing. It has to be a multi-pronged attack hitting all three C's.
Makes sense.
So to tackle camouflage, the physical barriers, we can try vascular normalization. Use low doses of anti-angiogenic drugs like bevacizumab, not to kill vessels, but to make them less chaotic, more normal.
Reduce that hypoxia and pressure, let T cells in.
Right, and for that stiff ECM wall, we can use enzymes like PEGPH20, which degrades hyaluronic acid, a major matrix component, or use LOX inhibitors to stop that collagen cross-linking, trying to loosen up the matrix.
Okay, opening the gates. What about waking up the immune system itself, getting that initial alarm going?
That's where targeting the CGS sting axis comes in. It's a critical pathway inside cells that detects misplaced DNA, like from a virus or a damaged cell, and triggers a potent antiviral pro-inflammatory response, including type I interferon.
Which tumors often silence.
Exactly. So researchers are developing ways to deliver sting agonists molecules that activate this pathway directly into the tumor. This basically forces the alarm signal, mobilizes those CDC1s, and gets the T cell response rolling.
Kicking the system back into gear. And what about the invisibility cloak, the MHC problems?
Epigenetic reprogramming. Using drugs that inhibit those silencing enzymes we talked about, like EZH2 inhibitors and tazemetostat, or DNMT inhibitors to cytobine.
Wiping off the molecular graffiti.
That's the idea. Remove the silencing marks to hopefully switch back on the genes for MHC components, or for those T-cell attracting chemokines like CXCL9 and CXCL10, make the tumor visible again.
Okay, that's camouflage. How do we counter coercion? The Hunger Games.
Metabolic interventions. We can try to block the tumor's fuel lines or neutralize its weapons, things like inhibiting glutamine uptake with drugs like teleglenistat.
Stop them stealing the amino acids.
Or block lactate's effects using MCT4 inhibitors. And a big target is that immunosuppressive adenosine. We can use antibodies like oleclumab to block the enzymes CD39 and CD73 that produce it.
Turning off the adenosine production line.
Right, trying to rebalance the metabolic battlefield.
And what about that neural connection, the hidden wires?
Neural interception, like we discussed, using beta blockers like propranolol to block the sympathetic stand-down signal, or targeting the sensory side, maybe using drugs like Remachant, which is already approved for migraine, to block the CGRP receptor RAMP1 and prevent that exhaustion signal.
Using existing drugs in new ways, potentially.
Fascinating.
Okay, and finally, you mentioned this revolution in delivery.
Ah, yes, the biomaterials revolution. This is huge because getting these drugs to the tumor and keeping them there working synergistically is a massive challenge.
Right, the TME is hostile territory.
Exactly. So material scientists are engineering smart delivery systems, nanogels, polymer prodrugs, nanoparticles. They can be designed to accumulate in the tumor passively through leaky vessels, the EPR effect, or actively target specific tumor receptors.
Like little smart bombs.
Kind of. And they can be designed to release their payload only in response to specific triggers in the TME, like low pH or certain enzymes. But one really cool example is cell membrane camouflaged nanocarriers, CMNPs.
Camouflaged how?
They literally coat the nanoparticle with membranes from, say, red blood cells. This cloak includes proteins like CD47, the don't eat me signals.
So the nanoparticle itself uses camouflage to sneak past the immune system.
Precisely. It evades clearance, travels deeper into the tumor, and then delivers its cargo, maybe a drug designed to reprogram those suppressive M2 TAMs into pro-inflammatory M1 macrophages right inside the enemy fortress.
That's leveraging the tumor's own tricks against it. Okay, so wrapping this up, what's the big picture?
The big picture is that the cold tumor isn't just passively resistant. It's an active, adaptive fortress employing incredibly sophisticated camouflage, hiding, blocking access, coercion, suppressive cells, metabolic warfare, neural manipulation, and cytoprotection, resisting death, instant repair.
It's a multi-layered defense system.
Totally. And overcoming it requires an equally sophisticated multimodal attack plan. We can't just use one key. We need the whole key ring, maybe even a crowbar. We have to treat the environment, not just the cancer cell in isolation.
And figuring out which keys to use for which fortress. That's the jam.
Absolutely. And that's why technologies like spatial multiomics, which let us map where different cells and molecules are within the tumor, and AI machine learning are becoming so critical. They can help us dissect that complex TME heterogeneity for each patient.
To personalize the combination strategy.
Exactly. To understand the specific defenses of that patient's tumor and choose the right combination of tools to dismantle them. It's incredibly complex, but also hopeful. Now, we know things beyond the tumor itself, like our diet, sleep patterns, stress levels via that neural axis can influence whether a tumor's hot or cold. makes you wonder.
Mm-hmm.
Considering how interconnected everything is, what seemingly small everyday habit might we overlook that could actually be, helping a tumor stay cold? And looking forward, where do you think the next big breakthrough might come from? Is there a master switch, a single target that could potentially disrupt camouflage, coercion, and cytoprotection all at once?
Oh, that's the billion-dollar question, isn't it?
It is, and that's the question we'll leave you, our listeners, to think about today.